Welcome to our channel. After a long back, I be here to discuss a very interesting topic, that is history of microbiology. Okay, let us move on to the topic, history of microbiology. How the microbiology will arise. Before going to that, father of microbiology is Anthony van Leeuwenhoek is the father of microbiology. He invented the first bacteria. So he called as the father of microbiology. Father of modern microbiology is Louis Pasteur, called as a modern microbiology. He is after the Anthony van Leeuwenhoek. He followed the guidelines of Anthony van Leeuwenhoek to proceed the work related to bacteria. Microbiology. What is meant by microbiology? First of all, we want to split this word into three parts. Micro, bio, logy. Micro means small, minute. Bio means life, the things having the life. Logy means study. Microbiology means the study is related to the small microorganisms. That is the microbiology. Microbiology is a branch of science that deals with the study of microscopic forms of life, that is microorganisms. Study of microscopic forms means the organisms which we cannot see with our naked eye, that can be seen with the help of a microscope or a compound microscope. Study of those minute organisms is called as a microbiology. That is nothing but a microorganisms such as bacteria, algae, fungi, like that, viruses. Next one, microorganisms are present in everywhere in the nature. Microorganisms are present in everywhere in the nature. So those microorganisms are called as a omnipresent. Omnipresent means it can present in everywhere in the place. Such as the floor, wood, everywhere in the surface the microorganisms are present. These microorganisms have great effect on other life forms. The microorganisms are very useful to the other living organisms like human beings. Without the microorganisms we can't live. Some microorganisms are very useful to us in making so many products. Like milk products also coming with the help of so many bacteria. We can't live without any bacteria. At the same time, some bacteria are causing the diseases to the other life forms. The microorganisms show a wide range of activity and diversity. Okay? Next, first of all, we go to the father of microbiology. About father of microbiology, who is Anthony van Leeuwenhoek, 1630 to 1723. He invented the microscope. He is the first scientist who invented the microscope. So he called as a father of microbiology. Anthony van Leeuwenhoek is a microbiologist and microscopist. Leeuwenhoek from Delft and Holland. He is the first scientist who described the bacteria. He is the first scientist who described the bacteria is called as a bacteria by Anthony van Leeuwenhoek. No other scientist cannot describe bacteria before the Anthony van Leeuwenhoek. So he called as the father of microbiology. He invented 419 lenses and 247 microscopes. On 7th September 1674, he described very little animalcules. Nowadays we call that the protozoans. On 7th September, he described the very little animalcules. Animalcules, nowadays we call it protozoans. Protozoans, euglena, paramecium. Paramecium is comes under the protozoans. It is called as a slipper animalcule. Other name of the paramecium is slipper animalcule because its structure is like a slipper. So we call as a slipper animalcule. He introduced the very little animalcules on 7th September. On 16th June 1675, he described very little animalcules of different shapes, sizes and motility. On 7th September, he described the little animalcules. On 16th June, he described the different shapes and sizes and motility of that animalcules. Anthony van Leeuwenhoek first described bacteria on 9th October 1675. To Henry Oldenburg, Secretary of Royal Society. He introduces bacteria on 9th October to Henry Oldenburg, Secretary of Royal Society. In 1683, he described the three shapes of bacteria, that is, broad, spherical, spiral shaped. In 1683, he described the bacteria shapes, how the bacteria will it may be rod shape or spherical shape or spiral shape. He made simple magnifying lenses with magnifying powers of 42 to 270 times. He made a magnifying lenses with magnifying powers also with powers 42 to 270 times. Anthony van Leeuwenhoek was first who reported accurate description of various types of bacteria to Royal Society. He is the first scientist who described bacteria and also who reported accurate description of various types of bacteria. About the types of bacteria also, he is the first scientist who described the types of bacteria and the shapes of bacteria to the Royal Society. Next one, Louis Pasteur, called as a father of modern microbiology. 1820 to 1895. Louis Pasteur, widely regarded as the father of modern microbiology, and he is the one of the three main founders of bacteriology together with Kohn and Robert Koch. Louis Pasteur is the one of the scientists who is the father of microbiology, and he is the main founders of bacteriology. He also works related to bacteria structure, bacteria functions and bacteria shapes after the Anthony van Leeuwenhoek together with the Kohn and Robert Koch. He is a French chemist, biologist and microbiologist, Louis Pasteur. He found out the fermentation process and these were carried out by the different microorganisms. We all know that the fermentation process is first invented by the Louis Pasteur, and this fermentation process is carried out by the microorganisms. These sentences are also stated by the Louis Pasteur. Fermentation means it's a process in which the heating up of a its fermentation process is always related to the microorganisms. It involves different types of microorganisms. In dairy products or in milk factories, they involve so many microorganisms. Okay, he noticed that the fermentation process is also carried out by the different microorganisms. He was a trained chemist. The studies on fermentation increased the interest in microorganisms. When he was studying on the fermentation process, he interested in studying also the microorganisms, about the microorganisms. By studying fermentation process, he also interested to study about the microorganisms. He developed the pasteurization process. Pasteurization process is used to remove the unwanted microbes in the industry. It is a process which are widely used in industry, used to remove the unwanted microorganisms. Suppose we are preparing the useful products like milk products, any milk products. If we need some important microorganisms. Along with the useful microorganisms, the unwanted microorganisms are present in that. We use the pasteurization process. We remove the unwanted products. We are only using the useful products, so we almost get the very desired product. Along with the useful microorganisms, the unwanted microbes is also there. Means the product is not a desired product and it does not contain the more shelf life. Pasteurization is a process of heating or boiling the liquid medium at 63 degrees Celsius for 30 minutes. It's called as a pasteurization. Pasteurization is the process of heating with a high temperature like 63 degrees Celsius at 30 minutes. It's called as a pasteurization. Nowadays, this pasteurization technique is widely used in the dairy industry. Mostly, the pasteurization technique is widely used in the dairy industry for preparing the useful products related to milk products. During his fermentation studies, he discovered some facts. The Louis Pasteur is very interested in studies regarding to fermentation process. He noticed some points while studying in fermentation processes. Fermentation is related to the microbial activity. Okay, fermentation is a process which is involved in the activity of so many microorganisms. Without the microorganisms, the fermentation process is not work properly or is not there. The microorganisms used in the fermentation may be bacteria or yeast. Mostly in the fermentation process, we use the bacteria and the yeast as a microorganisms. In each fermentation, a selective microorganisms and specific conditions are required to obtaining a desired product. In fermentation process, only the selective microorganisms are required and specific conditions. At some conditions, specific conditions like at 63 degrees Celsius, at the constant pH, and the desired microorganisms, only the useful microorganisms. Under these conditions, we obtain the desired conditions. In the fermentation process, if we are using the unwanted microorganisms under different conditions, so under these conditions, we are not getting desired product. Only the selective microorganisms and specific conditions are needed for getting a desired product. During his study on butyric acid fermentation, he discovered anaerobic bacteria. During his study on butyric acid fermentation, he discovered the aerobic bacteria. The bacteria which can live by using oxygen is called as the aerobic bacteria. In 1981, Louis Pasteur discovered vaccine for cholera, anthrax and rabies. In 1981, Louis Pasteur discovered the first vaccine for cholera, anthrax and rabies. Next scientist is Robert Koch. Along with the Louis Pasteur, the three founders for the Louis Pasteur work together with the Robert Koch scientist and Kohn. Robert Koch, 1843-1910. Robert Koch was a German bacteriologist. He is also one of bacteriologists. He isolated causative agents that are responsible for causing the diseases such as anthrax, tuberculosis and cholera. Before the Robert Koch, Louis Pasteur discovered the vaccines for anthrax, cholera and rabies. Robert Koch, after isolate the causative agent, which organisms or which bacteria are responsible for causing the anthrax, tuberculosis and cholera. He isolate the causative agent. Here some agents are there for causing anthrax, Bacillus anthracis, 1876. In 1876, he introduced he isolated the causative organism, that is Bacillus anthracis can cause the anthrax disease. Tuberculosis by Mycobacterium tuberculosis in 1882. Cholera, Vibrio cholerae in 1883. Further, Robert Koch invented the stick plate technique. Stick plate technique is the one of the technique which is used for the separation of the microorganisms or any particles relating to their size. Okay? Relating to the size of the microorganisms, we are separating those microorganisms by using the stick plate technique. introduced the use of gelatin as a solidifying agent in the nutrient medium. He used the gelatin is a substance which is used as a solidifying agent in the nutrient medium. Nutrient medium is a solution or a liquid in which the culture of microorganisms can occur. We can grow so many useful microorganisms by using the nutrient medium. He introduced the gelatin is used as a solidifying agent. He introducing straining techniques. Robert Koch introduced the different straining techniques also introduced. Next one, Robert Koch postulates. Some postulates are introduced by the Koch. That is, microorganisms that is responsible for the disease should be constantly present are found in the lesions of the disease. The microorganisms which is responsible for disease. For example, anthrax. Anthrax is a disease caused by the some bacteria. Constantly, that the causative bacteria will be present in the or found in the lesions of the disease. Lesions means the cell or a tissue which is affected by a disease. In that cell, the causative agents are also the microorganisms are also present in that lesions only. So it may cause the disease. This is the one Koch postulate. Pure culture of organisms can be isolated from the lesion of disease. Pure culture of organisms. In the lesions, the causative agents of so many diseases are present. And that lesions, we can extract the pure culture of organisms also can be extracted or isolated from the lesion of the disease. Inoculation of pure culture is suitable laboratory animals. Inoculation of pure culture in suitable laboratory animals also can be done. These are the Koch postulates. Next scientist is Joseph Lister, 1827-1912. Joseph Lister was an English surgeon and he developed antiseptic surgery and pure culture technique. He is an English surgeon and developed the antiseptic surgery and a pure culture technique. Carbolic acid, a chemical used as an antiseptic, proved by the Joseph Lister. He proved that carbolic acid is a chemical that is used as an antiseptic. He is the first scientist who proved carbolic acid is used as an antiseptic. Okay.